So you know what I am talking about, right? I am talking about troponin, okay? Troponin means what? Let's again go back to the F-actin, okay? G-actin, put together, okay? And then 2 rows, okay? And then the twist form a helix, okay? And then each one has a site where the myosin head can bind, okay? And then there are 2 more proteins. One is called as tropomyosin, which is a double helix here. There are 2 of them, one is here, and they go all along and they are occupying a position such that on each G-actin, where the site where myosin is supposed to bind is occupied. So, is the binding site on the G-actin molecule available to the head of the myosin? No, it is not available. Okay. Then there is yet another molecule which is called as troponin, yet another protein, troponin. What do you call it as? Troponin. Now, this troponin is actually made up of 3 subunits. How many subunits did I say? 3 subunits. One binds to tropomyosin. So, troponin binds to tropomyosin. 3 subunits, one of them binds to what? Tropomyosin. Good. The second one binds to actin, okay, and the third one binds to calcium whenever it is available. So, what am I talking about? I am talking of troponin made of how many subunits? 3. One of goes to actin, okay, one goes to tropomyosin, and one is there for the calcium ion to bind. Now, how this works we will see shortly. Okay. Now, we are looking at the same thing. We are looking at the F-actin is there and tropomyosin is there. If you see carefully here, there is a circle, and the circle there is that dark patch there. Dark patch is the site where the myosin head is supposed to bind, but it is covered. You can see all this that tropomyosin is covering. But whenever on the troponin the calcium binds, something very interesting happens. What do you expect? The tropomyosin to move away. And once the tropomyosin moves away, then the binding site is exposed, and then it is just ready for the head of the myosin molecule to bind with its site. Are you getting the argument? So, who is inhibiting the interaction? It is the tropomyosin. Okay. Why would tropomyosin be displaced? Okay, it would be displaced because troponin will pull it away. Why would troponin pull it away? Because calcium needs to activate it. Okay. Now, why would calcium activate it? We'll see that shortly in a couple of slides. So, now we are ready to see the entire what they call, cross-bridge cycling. What do I mean by cross-bridge cycling? Now, I am trying to animate something here. This is myosin. What is this? Myosin. And this is the head of the myosin. Okay. And this is the actin. Are you okay so far? And the Z membrane is somewhere there. Okay. And this is actin. It is going all the way and sticking on to the Z membrane. Are you okay so far? It is very funny. Okay. But this is the Z membrane, this here, and this is the myosin molecule. Okay. When calcium becomes available, okay, and then the tropomyosin moves away. Are you with me? Tropomyosin moves away. Okay, then it can bind. So, what has happened is on the actin, there is one G guy sitting here. On that there is an active site. Okay, where the myosin head is bound. Okay. Now, once it is bound, it undergoes through a stroke. Power stroke. What do I call it as? In power stroke, it moves this way. It moves this way. It moves this way. Now, this actin molecule is sticking onto this Z membrane here. It will pull the actin towards the Z membrane. You got the question? Now, I am going to show the same phenomena I will try to explain it in this slide. So, let us go to the top, and in the top we have a position. Okay. Now, we remember at the top we have here is a myosin filament. Good. In the myosin filament, we are now focused on a single myosin molecule, and of that molecule, can you identify the 2 heads there, please? 2 heads are there, and out of the 2 heads we find that one head is already sticking on to the actin molecule on its binding site. So, they are already together. Now, this is about step number 1. We will start here and come back to the same point, which means we will complete a cycle, which we will call as a power cycle. Okay. It is a transient state. It's just holding on further for a very little time, and this position is called as a Rigor state, R-I-G-O-R. Okay. This Rigor is very important, called as, so that state is called as Rigor state. Which part of the head? Okay, the myosin has a cross bridge. On the head there is the site. So, this protein which is sitting here has an affinity for the G protein site, and they are together. Good. Now, before I go ahead, I just want to ask one thing. If this were the tail of what? The myosin molecule. Tail, then neck, hello, and neck, and then the head. Okay, and the head is bent. And I'll draw your attention to the fact that the head is bent at an angle which is less than 90 degrees, about an acute angle. Are you okay so far? It's an acute angle, mind you, it's an acute angle. This is an acute angle. So these are the two heads, this is the neck, and the neck has yet another peculiarity. You know, 4 more proteins are sitting on the neck. Hello, what do you call them as? Very good, very good. What do you call them as? Light chain, light chain. So you have four light chain molecules and a bend, and somewhere a little behind that is a site where ATP can bind. ATP can bind. And what is the peculiarity of the part of the protein which is sitting there in the ATP binding site? It is ATPase. What is it? It is ATPase. Okay. So just imagine how big the molecule must be, how subunits must be. So at one end it has got that protein can combine with another protein which is sitting on the which is on the F actin there. It can bind in another part where it can combine with the ATP molecule. And now we have an interesting situation in which the ATP molecule combines. Well, it will combine on this side, which is the ATP binding site. Now with every little thing, there is going to be a configurational change in the protein molecule. Now, what configurational change is? Would you expect? What difference do you find between this state and this state? Number one, ATP has bounded its site. Are you okay so far? Now you see a very interesting thing has happened. What is it? It is unbound, dissociated. What has dissociated from what? Tell me. Myosin has dissociated from actin. So if you don't provide ATP, the dissociation won't happen. So as if the moment ATP combines at its site, there is a tremendous change on the charge in the molecule. As a result of that, it's a slightly different molecule and it loses its affinity and you get this particular stage. And then as you can imagine, the ATP is bound, it is bound to its site, and that site is ATPase. ATP is going to break away the molecule, and soon thereafter you will have a situation in which, what do you have here? Can you tell me? Look at that cartoon and tell me. Hydrolysis. So ATP is broken down into ADP and P1. Okay, Pi. Are you okay? Are you okay so far? Okay. So it has been, so the enzyme, as a result of hydrolysis of ATP, you have ADP and P. Are you okay so far? Then in the next, look. The neck is still an acute angle. The neck is still an acute angle. The neck is still an acute angle. Are you done so far? Hello. Now let's move on. In the next step, you see it binds to here as a result of the hydrolysis. Then you have a similar situation in which very interesting phenomena is happening. I have been harping on the acute. Now if you see carefully, it is no more acute, but it has come at a right angle stage. Okay, means it was so much bent. Unfortunately, my wrist can't bend so much. Okay, it's much more bent, and then it becomes so this whereas this angle was acute, this is more or less so you can see that arrow, and this is called as a cocked position. It is like you have charged the gun, you have charged the molecule, and this charged molecule here, means actually what has happened is the energy from the ATP now has been taken by the molecule. Which molecule? The myosin head. Okay, and the myosin head has bound. It has taken the energy, and the next step is can you tell me the interpretation of this diagram there? Pi goes away, and the moment Pi goes away, then the cocked molecule again goes from a right angle position to the acute angle. Can you see in that image, please? And in that process, this molecule is pushed towards the Z membrane. So Z membrane is somewhere here. This molecule is anchored on the Z membrane, and as a result of the molecule being pulled, the actin molecule is the two Z membranes on the opposite sides will be. Am I making sense? Hello. Are you okay so far? Sure. We are good so far? Okay, this one also? Just not much has happened. Lot has happened. Anyway, the ATP is bound there, okay, and then the ATP has been acted upon by the enzyme, okay, and that enzyme is an integral part of the myosin head, okay, so far. And then so here ATP has been converted to ADP and P, and then here you have as a result of this hydrolysis, now the head of the myosin molecule is ready to bind. It binds. Okay, so far. Then as a result of that, then the next step would be the P was still there, but the P goes away. Okay, so far. Once P goes away, okay. So at this stage, can you see that tiny circular arrow there? which means from the acute angle it has gone to, so it was very much bent. Now it can now from acute it can go to right angle, and now once the P goes away, it goes into what you call as the power stroke. Means as you go from this step to this step, again as you go from this step to this step, myosin molecule is pulling the actin molecule by a distance of about 10 nanometers. Means chemical energy is converted into mechanical energy. Did you get the message now? Very simple and straight. Are you okay now? Okay. Now, so after that, then in the next step, the ADP was here. Hello. Now the ADP goes away. Okay. Now once the ADP goes away, but it still remains bound. It still remains bound. It still remains bound. It will release, it means it is in the Rigor state, and it will go away only when the next ATP molecule comes. Means for a single myosin head to interact with a single site on the G actin and to pull it once, you need one molecule of ATP. And then this is how we say that the cross bridge. So this is one, and on every cell there are thousands of cross bridges, thousands of actin molecules, and all put together they will generate the force, which is nothing but the contraction of the muscle. Now look at the cartoon and tell me, if I withdraw the ATP from the system altogether, artificially, imagine for a second, if I withdraw, what will happen? It will stay, absolutely correct, it will stay in this position. It will stay in what position? It will stay in Rigor state. Are you with me? Are you with me so far? After the death of a human being, after a period of about 40 minutes or 60 minutes, the body becomes absolutely stiff. And that state is called as rigor mortis. Mortis refers to death. Rigor. That rigor happens because after the death, the ATP is no more available. Because ATP is no more available, the myosin molecules remains attached to the actin molecule for a very long time. Okay, and that stage, and then eventually, however, after the death, about 1 hour, 2 hours, 3 hours, even whole protein system, actin myosin molecules, they start breaking down. Once they start breaking down, then the body loosens again, and which clinical people or medical people say that rigor mortis has set in when the body is stiff. Okay, you might have seen this in murder thrillers, when the body is stiff, and then after about 3 or 4 hours, the rigor mortis passes away. Whereas in the living condition, the rigor is a transient state. It comes and it goes because ATP comes and binds. Okay, but had ATP not been there, we would have. So I'm introducing you to another word, rigor and rigor mortis. Go ahead.
So could you go to the previous slide? So, but for rigor mortis to happen, there has to be calcium present inside the sites, right? Because only then the actin binding site will be available.
At the time of death, whatever was available, depending on whatever interaction has taken place into actin myosin. Okay, it's not possible that there is no interaction happened at the time of death. Some will be there. Okay, all of them, whatever is there, okay, they will sustain.
Why does power stroke has to be coupled with the release of inorganic phosphate?
Why does say that again?
Why does the power stroke have to be coupled with the release of?
Oh, it is the you have to see these in terms of the configurational changes that is happening in the proteins on the head. Okay, and it is of course it's a question of evolution. It has evolved in such a way that this is the way it happens. Okay, you can search for the logic. Okay, but then you can search for logic everywhere. Okay, which is very but because and you ask the question 10 times, the answer is in evolution. Okay, this is the way the things have evolved. But if you will find some logic. I mean, after all, but the stream of the ideas is that for the configurational to change from one to other. Okay, this ATP comes, it undergoes this, then enzyme acts on this there. So it is. Yes.
with with uh, with cycle on cycle, as contraction happens in the Z membrane,
Yes. Yes.
the spring action of the titan continuously gets more and more compressed, right?
It does.
Every single new ATP that binds, and when the head detaches from the actin, that spring will want to decompress and expand the sarcomere again.
The answer to that question is that spring works much better when you pull it. When you pull it, so muscle is 5 inch long. Okay, if I pull it, the spring will stretch, and the spring will try to bring it to 5 inches again. Okay, so there is a spring in front of me. That spring is 5 inches long. I'll pull it to 6 inches. That spring will try to bring me back to 5 inches. But when I compress it there is not so much load on that. Am I making sense there?
So when you have a muscle that is compressed, a flexor that is compressed,
Yes.
when the corresponding extensor contracts, and then you have the flexor expanding, how does the cycling work then?
Okay. No. Okay. I'll come to that when I talk about acetylcholine. So what you are saying is what happens when the muscle relaxes? Yeah. When muscle relaxes, there is no acetylcholine coming. How it is relevant, I'll talk to you in 5 minutes. There's no calcium available. Very simple. Very simple. The answer to your question is when no calcium is available. When no calcium is available, the muscle relaxes. That's it. All you need to do is either provide calcium or don't provide calcium. If you want to contract, provide calcium. Calcium. Is that message fully taken? So when you do this and this when this muscle is relaxing, that's because there is no calcium available. So no cross bridge, no actin myosin interaction. Done? Great. Yeah.
Just a follow-up question on that. Does it mean that the sarcomere won't revert back to its original position by itself? Like the extensor has to bring it back?
Which one?
Like
No. That you see, that's where the other proteins come into picture. Okay, it is okay. What you are saying is what happens when there's no calcium. When there's no calcium, then there are no cross bridges. Okay, and when there are no cross bridges, the muscle will go back to its original length of each sarcomere, and for that other proteins play an important role. Okay, bring it back. They have to go back. They have to go back. Otherwise, how will you again contact it for the next time? So calcium is very important. Okay. So calcium binds where? Okay. Listen to this. This is very, very interesting. I am talking about troponin molecule. What am I talking about? Troponin. In the troponin molecule, there are 3 subunits. Done. Okay. The real excitement lies in that part which binds with calcium. Okay. Others are also important. Otherwise, protein chemistry people will kill me. The part that binds with calcium is very important. Now that molecule, single molecule, single what molecule? Single subunit of troponin. It is supposed to bind to calcium. Single molecule binds with 4 ions of calcium. How many ions? 4 ions of calcium. How many ions? Well, there are n number of molecules. So all of them, but for a molecule only 4 ions of calcium can combine. So you have a protein molecule, one of the 3 subunits of the troponin, big molecule, and there are 4 sites where calcium can combine. Now imagine that molecule at the moment to begin with doesn't have calcium. So all the 4 sites are vacant. Are you okay so far? Now one calcium combines. One. I'll go steadily. 1, 2, 3, 4. 1. When one combines, there is a change in the configuration of the molecule. The first one combined with affinity, certain affinity, as happens between 2 bodies, 2 molecules, calcium ion versus what, and I'll call that affinity as X, whatever it is, X, some value, I don't know, X value. But because one ion of calcium has combined, okay, affinity has changed. As a result of that, the second calcium ion combines with affinity which is 2X. Hello. So one calcium ion combining with the protein changes the affinity for the next site. One has already combined, next site affinity is more. So, when the second calcium ion combines, it influences the affinity for the third, now it shows 4x affinity. And the next one may be 10x, 20x, I don't know. This is a phenomena which is very common in biology, one of the amazing phenomena which have evolved, and we call it as cooperativity. What do you call it as? Cooperativity. I will again talk about this phenomena when we talk about how the hemoglobin molecule combines with oxygen. That molecule, so many enzymes show the phenomena of what cooperativity means. Once it starts going in one direction, its efficiency is increased many folds. This is a very interesting animation video on the YouTube. It tells everything what we have done. Okay, so I'll strongly recommend that you spend a few minutes and okay. We have actually seen this image, but I am putting this once again. So, what are we seeing here? We are seeing a direct correlation between the actin-myosin sliding over and work being done. Okay. Now, I ask you one very simple question. Look at the degree of the overlap between actin-myosin filaments in the image number 1, 2 and 3. And what is your observation? It is what? It increasing. Can somebody please explain to me as to what would be the hidden meaning there? More overlap means what? More overlap means what? You are right. More overlap means more opportunities for the myosin head to combine with actin. See if look. See this is the myosin molecule in my traditional myosin. This is actin, and in this there are say 100 myosin heads. Are you okay so far? 100 myosin heads. I am just taking arbitrary figures. 100 myosin heads. Now how many will be there now? More, 200, maybe 300. And how many will be there now? Nil. You see, if there is no overlap between actin-myosin filaments, the muscle is useless. It can't contract at all, because the force of contraction or power of contraction is dependent on the number of, directly dependent on the number of how many heads can combine with how many molecules, how many of their sites on the actin molecule. Are you with me? Yeah.
Are it the myosin which only present in the ends?
Which one? They are present only in the
Ends of the...
Yeah, on the two ends.
So even if there's a great amount of overlap, only that part can bind to the actin.
I didn't get your statement. Can you say that again, please?
So because the myosin heads are only present on one end.
On two ends, no? I mean, yes. Ends, okay.
If the overlap increases, the
Yeah.
area of the heads, how much it overlaps with actin will remain constant after a certain extent.
It will. You see, after a certain limit, as in this case might be, you see, and once the overlap is complete, then no matter whatever you do, okay, there won't be any additional force. Means what? The amount of force that a muscle can generate depends on how many opportunities are there for the head to combine with the heads of the myosin to combine with the actin molecule, plain and simple. Another beauty of the situation is that each head is on its own, if you can get my language. Means one myosin head and second myosin head, it has nothing to do with one another. Each myosin head, as long as you keep on providing ATP molecule to it, it will keep on beating its own, at its own frequency, it will keep on beating. Are you getting the argument? So it will go, so it will one actin, second actin, third, like a, it is, people often compare, in Kerala they have boat races, they pull the oars. Are you following what I am saying? Okay, they have boat races in Kerala, very interesting episode. You see, they just pull. So you just imagine that oar is the head of the myosin molecule, and you have to pull it. Okay, so but every oar is independent on its own, and as long as every oar gets an ATP molecule, it will go to the next available molecule on the actin and pull it as long as calcium is available. As long as what? Calcium is available. Now, how does the calcium become available is a point that we will try to sort out.
So, excuse me.
Yeah.
As you said that each head is independent, does it mean that they are not at all coordinated, or they are coordinated?
They are not coordinated. That's what I am saying. They are not coordinated. If a particular head doesn't get ATP, it won't function. But if the next guy gets it, it will function. I mean, the cell provides ATP, the cell provides everything, but ultimately it is just left to the chance of a head. If you get ATP, thy shall function, otherwise you cannot function. And one has nothing to do with the other. There is no central control. That answers your question? Oh, it can, any of can combine. Any can combine, any can combine. If it finds an actin there, if it can bind there, it will work. And often it works.